Most people have some familiarity with genetics, but the newer field of genomics is still mysterious to many. In this video, I'll provide an overview of what genomics is. By overview, I mean that although I can't be comprehensive in one short series, I can give you the gist of what genomics is all about. I can also set the stage for future videos in which we can go deeper to look at the ways that genomics is likely to change your life. Let's start with an overview to give you a sense of the big picture, both what genomics is and how I'll approach introducing it to you. There are two ways to think about genomics. First, genomics is a study of genomes. A genome is all the DNA in some species. This is perhaps the easiest way to think about genomics, and it's the most common way it's introduced in molecular biology classes. The second way to think about genomics is that it's basically genetics done on a vast scale, meaning that instead of studying one gene at a time, you study many genes at once. There is much to learn from both approaches, but here I'll take the second because it's a little more intuitive for people without a strong background in biology. I'll make a companion series, however, in which I'll introduce genomics from the other approach, sometimes called the molecular genetics approach. That series will be complementary with this one, and both together will greatly increase your understanding of genomics. As I said, genomics is basically genetics on a huge scale, so we have to start by introducing genetics to make clear why the switch to genomics was so necessary and important. Of course, genetics is a big field requiring an in-depth introduction of its own. This cannot be done in one video, but I can quickly provide enough background for you to be able to appreciate the importance of the transition from genetics to genomics. Genetics is the study of inheritance. We all know, for example, that couples in which both parents have red hair are far more likely to have red-haired children than are couples with one or no red-haired spouses. Most people also know that the reason is that red hair has a genetic basis which is passed to the offspring via what are commonly called red-haired genes. I stress the word commonly because in biology we have a more technical way of describing this, such that we would say that there are alleles, or variations of a gene, that code for, which is to say, cause red hair. The way we learn about genetics in high school stresses things like alleles for blue eyes or red hair, and the mechanism we learn is called Mendelian genetics. This figure is a good summary of Mendelian genetics, but we don't actually need all this detail for our present discussion. For our purposes, we can simply say that Mendelian genetics is the genetics of cases in which differences in a single gene cause phenotypic differences. Phenotype may be new terminology. It refers to the state of the trait we are focused on. Eye color is a trait, and blue or brown are phenotypes. Again, for Mendelian traits, the phenotypic variation we are interested in is controlled by variable forms, or alleles, of one gene. Here we see a chromosome with two different alleles for some gene. The position on the chromosome where the gene or anything else lies is called a locus, by the way. I may use this terminology later. Variation refers to different sequences of bases, or nucleotides, in the DNA that makes up the gene. The bases are the familiar A's, T's, C's, and G's that make up DNA. I started by saying that the way we learn about genetics in high school stresses Mendelian genetics. The reason I stress this is because it's misleading. In reality, Mendelian genetics is a tiny part of genetics. Moreover, many things that we're taught are Mendelian are actually not. I mentioned hair and eye color and the alleles that code for red hair or blue eyes, for example, because these are commonly known examples of genetic variation, and they're sometimes taught in the Mendelian manner in introductory classes, particularly in high school. But in reality, many genes contribute to your hair or eye color. What makes eye color seem Mendelian is that some of these genes have a very strong effect relative to the others. For example, there are many shades of every hair and eye color. Further, red-haired parents do not always produce red-haired children, even though they usually do. So there's more going on than just a few alleles from one locus. Unlike in this graphic, eye color is not a simple trait that you can predict with Mendelian genetics. There are many genes that influence it. We don't have to understand all of this now. I'm only trying to get across the fact that genes usually work together in groups, even for relatively simple traits like eye color. This is a gene network associated with cancer. Each colored circle or diamond is a gene, and the lines between them mean that they interact. Most traits have a genetic basis of comparable complexity, meaning that all of these genes would interact to determine the state of the trait. Even for simple traits that are purely Mendelian, it is not the case that the relevant genes work in isolation. Imagine all the genes in a network, but one are fixed. That is, the genes have no meaningful variation. Each gene does its job the same way in all individuals. Imagine further that one variable gene, the lit up one in the middle of this pic for example, has two alleles, one of which is broken with respect to what the gene is supposed to do. This simple situation will cause a Mendelian phenotype. It could explain the color of a flower, for example. Imagine red is a normal color, and that red occurs when all the genes in the pathway are functional. But if one gene is broken, then no color is made and the flower is white. We therefore have two alleles that correspond to two Mendelian traits, but flower color is based on this whole pathway. So Mendelian genetics is simple, but what is called molecular genetics, which includes the whole genetic basis of the trait, is complex. Further still, the genetic variation underlying phenotypic variation is usually far more complex than a single change in one gene causing a Mendelian trait. In fact, that is the simplest form of genetic variation, which is why it was discovered first. In reality, many genes in the pathway might vary in complicated ways that give rise to a complex distribution of phenotypic variation, such as those we see in different human populations for traits such as hair texture or facial features. That's enough basic genetics. I think we're ready to introduce genomics. As you can tell from our discussion so far, to understand genetics deeply, you have to identify many genes, figure out what pathways they are part of, and determine how they fit into those pathways, or in other words, what their jobs are. You also have to map out all the variation for each gene. And pathways can have hundreds of genes. Further, a single gene can be in many pathways. Moreover, pathways are often connected to one another. We don't need to understand all of this for now. We just need to see how unbelievably complicated genetics is and how much information you need to understand how genetic systems work. In the past, it was very hard to identify a single gene with variation important for some trait. It was even harder to sequence the DNA for that gene. Sequencing refers to figuring out the sequence of bases in an allele, the order of A's, T's, G's, and C's. In fact, everything was hard and expensive in genetics. A person might do a whole PhD, 5 or 6 years of work, just figuring out the sequence of one gene and learning one or two simple things about how it worked. Because of this, our knowledge of genetics moved relatively slowly for many decades. I say relatively slowly because genetics is a huge field, with thousands of scientists all over the world working away at their particular problems. So even if the field as a whole was making good progress, a single geneticist was usually moving forward painfully slowly. All that changed when we developed the ability to sequence DNA at a fast rate for an affordable price. A budget that allowed you to sequence one gene in the 1990s will now allow you to sequence all the DNA in an organism. In the genomics era, a genetics PhD student doesn't study just one gene anymore. Rather, they might try to trace out the entire pathway underlying a trait and identify all the variation for each gene. But how that is done is too much for this video. Suffice it to say, to do things like that, you need a genome. So the first most central step in most genomics work is to get a genome for your species. For most of the rest of the series, we'll talk about how we get a genome. In other words, how we figure out the genetic sequence for all the DNA in a species. We'll conclude, though, with a key application that makes use of the genome to do genetics on a vast scale. I'm not going to go too much into the molecular biology or the details of the sequencing technology, or in other words, how the machines work, because that's not necessary for understanding what genomics is all about. The basic ideas and concepts are enough. In the old days, I would extract, which means to isolate and purify, a stretch of DNA containing my gene of interest, and then use Sanger sequencing to sequence it. Sanger sequencing was developed in the late 1970s. It was accurate and a game changer, but it was slow. The sequence I would get would look something like this. This is part of the sequence for the famous insulin gene. The sequence of nucleotides, the A's, C's, G's, and T's, is turned into a sequence of amino acids, which folds into a protein important for regulating blood sugar. Sanger sequencing can sequence up to about 1,000 nucleotides in a row. Genes vary in size from about 100 to 100,000 nucleotides in length. The basic point is that a single gene may need many runs of the Sanger machine to sequence all of its length. A genome is much bigger than a gene, making sequencing them prohibitively difficult with Sanger sequencing. For this reason, early genomics mainly focused on bacteria. Bacterial genomes are often only a few million bases long, meaning that a number of sequencing runs in the low thousands might be enough to get a genome. That sounds like a lot, but it was feasible. But animals and plants have far larger genomes. Insect genomes are commonly hundreds of millions of bases long, although they range up to several billion bases. Plant genomes vary even more dramatically, from tens of millions to more than 100 billion bases. Humans, by comparison, have about 3 billion bases. Sequencing a genome of 1 billion bases, if you can only do 1,000 at a time, means at least 1 million sequencing runs. In practice, you would need many more than that even, because of issues with assembly. Take a single gene 10,000 bases long. We can only sequence 1,000 bases at a time. Therefore, we have to break up the DNA of the gene into pieces, sequence them, and then put those pieces back together like a puzzle into the full gene sequence. That is called assembly. In this example, you might think that we would need to break the gene into 10 pieces, because that would give us the 10,000 bases of length. But to do assembly, you have to line up the pieces to figure out which match. To do this, you need a decent amount of overlap between the pieces. So in practice, you cannot just take the total length and divide by the number of bases that you can sequence at a time. There is a balance between sequencing the least amount of DNA possible and still having enough overlap to ensure accurate assembly. For genome assembly, as we'll see in part 2, where we talk about this in more depth, this is sometimes a very complex task. So genomics in the old Sanger sequencing era was limited mostly to bacteria and a few very important species such as humans. The Human Genome Project, for example, was done mainly with Sanger sequencing, but it took an enormous amount of time, money, and energy, and many people working on it. The human genome was completed in the mid-2000s, which is about the time that what is called next-generation sequencing was developed. Although genomics existed prior to next-gen sequencing, it really took off once these machines became available. We'll learn about all of that in the next video. I hope you stay tuned for part 2. If you enjoy this content, please like and subscribe. See you next time.